Hello, my name is Sarah Dickerson. I'm a scientist at Horizon Discovery, and I will be presenting this poster on CRISPR-A using synthetic guide RNA. So synthetic guide RNA is beneficial in many respects. For example, it allows for short-term expression and enables you to avoid cloning, which are some of the challenges associated with using express guide RNAs. CRISPR can be repurposed from a gene editing tool to a transcriptional regulation system. A nuclease deficient mutant of Cas9, known as dCas9, does not cleave the DNA, but retains the ability to bind guide RNAs and localize to the target DNA. When dCas9 is fused to a transcriptional regulator, it can activate transcription, commonly referred to as CRISPR-A. Several CRISPR-A systems have been developed, including SUNTAG, SAM, and VPR, which we show here. Initially, researchers used express single guide RNAs for CRISPR-A, and in data not shown, we have found that the synthetic two-part CRISPR RNA and tracer RNA system can also be used to activate transcription with CRISPR-A systems. Here we investigate the use of a 5' end modification of the CRISPR RNA and the 3' end modification of the tracer RNA in performing CRISPR-A. We wanted to see if these modifications would have an effect on activation, so we compared modified CRISPR RNA tracer RNA, which is shown in dark green, to unmodified, shown in light green, for 4 different genes, and then measured transcriptional activation via RT-qPCR. Here we show transcriptional fold activation for an individual CRISPR RNAs as well as the pools of 4. Modifications allow for a dramatic increase in activation. Additionally, the pool of 4 CRISPR RNAs also greatly enhance activation. For example, there is a more than 30-fold increase in activation of titin with modified CRISPR RNAs pooled compared to unmodified CRISPR RNAs. To determine how synthetic guide RNAs compared to express single guide RNA and lentiviral expression, we compared the 3 methods for 4 gene targets and report that synthetic CRISPR RNA tracer RNA result in gene activation comparable to expressed single guide RNA. Synthetic CRISPR RNA and tracer RNA can also be used with the SAM system. SAM is unique in that it uses a scaffold tracer RNA with an MS2 aptamer sequence that recruits transcriptional activators. We found that the transcriptional activation using either the SAM or the VPR system for 6 genes was comparable. There was no consistent trend across all genes. For example, transcriptional activation by SAM was higher for titin, whereas transcriptional activation for VPR was higher for IL-1 receptor 2. While most of the work presented here involves U2OS cells stably expressing dCas9 and the respective activators, we also demonstrate the effectiveness in using synthetic guide RNAs with dCas9 VPR mRNA. Furthermore, the advantage to using either puromycin dCas9 VPR mRNA or EGFP dCas9 VPR mRNA is that it allows for enrichment. It is important to note that the basal expression level of the target gene may impact the level of activation achievable. The bar graph represents transcriptional activation ranked highest to lowest for 23 different genes. The endogenous gene expression for each gene target obtained from RNA-seq data is shown on the right y-axis. We consistently found that genes that have very low endogenous expression levels are typically activated to a higher degree. For instance, endogenous expression of IL-1 receptor 2 is very low, so we are able to achieve a dramatic increase in activation with CRISPR-A. Synthetic guide RNAs are also effective for multiplexing in which more than one gene may be activated. We demonstrate that the level of transcriptional activation achieved is similar, whether a single gene, 2 genes, or 3 genes are simultaneously activated. Synthetic CRISPR-A, CRISPR RNA tracer RNA also enables pooled screening in an arrayed format. As shown here, using a cell line stably expressing dCas9 VPR allows for fast and effective screening. U2S cells stably expressing dCas9 VPR were transfected with the CRISPR-A library consisting of 153 cytokine receptor targets, where each well contained a pool of 4 CRISPR RNA tracer RNAs. targeting one gene. IL-6 secretion was monitored by ELISA, and then we were able to identify IL-6 regulators. And here we identify positive regulators of IL-6 in red and negative regulators shown here in blue. So the work presented here shows that synthetic guide RNA can be successfully used with CRISPR-A systems like VPR and SAM. Multiple CRISPR RNAs targeting the same gene can be pooled to achieve higher transcriptional activation of the target gene. We also demonstrate that synthetic CRISPR-RNA tracer RNA is ideal for simultaneous activation of multiple genes and is effective when used with dCas9 VPR mRNA. And finally, we showed that synthetic guide RNA can be used for high throughput arrayed screening applications. Thank you for listening, and please feel free to reach out if you have any questions.